start that. So for lecture for today, we'll spend some time talking about the intracellular traffic and protein sorting. And we should get through all of this today, and then we can at least start getting into the next lecture, which is signal transduction and secondary messengers. And then I'll stop a few minutes early and we'll do a quick activity at the end before we leave today. That will help me to see where we're at in the class and anything that might need to be clarified for next class. So where we are in this class in terms of topics, right? So we spent some time most recently talking about protein synthesis. And what we see here is now that we have the protein being made, proteins can be transported to various parts of the cell, and there's actually much protein transport that is taking place. There we go. And so as we learn, the synthesis of most proteins is going to begin in the cytosol of the cell. And the main exception here for eukaryotic cells is going to be some mitochondrial proteins. And these mitochondrial proteins, there's a set of ribosomes in a mitochondria. And so some mitochondrial proteins do not get synthesized in the cytosol of the cell, but rather in the mitochondria itself. But by and large, most proteins in the cell get produced in, or at least begin to be produced in the cytosol of the cell. And then what we see is that some of these proteins will remain in the cytosol, while other proteins will be transported to various organelles. Some proteins will be transported to the cell membrane and remain in the cell membrane, while others will be exported from the cell. In addition to this, there will be some proteins that are transported between various organelles that are found in a cell as well. So there's a lot of protein transport that is occurring here. An overview of protein transport in a cell is shown in this figure here. And the first thing I want to point out to you is not all of these organelles are necessarily found in every eukaryotic cell. So for instance, these things called plastids are found in plants. And you can cross that off of your slide here. So not that plants aren't important, but for this class here, we're not focusing on them. And then, for instance, another example where one of these organelles is not found is, red blood cells, right? Red blood cells do not have nuclei, for instance. And so, transport from the cytosol or between the cytosol and the nucleus in red blood cells doesn't occur, again, just because, frankly, they don't have the nuclei. So we see here there's a number of different organelles and a number of different types of transport that occurs between these. And as you can see here, looking at the slide, there are three general ways for proteins to be transported. And these are gated transport, transmembrane transport, and then finally there's one called vesicular transport. We're going to focus mainly on the last two of these today, but nonetheless, for gated transport, as you can tell here by the key here, this is occurring between the cytosol and the nucleus. And this occurs through some pores in the nuclear membrane. And what happens in these pores is that some small molecules can freely diffuse, in and out of the pores. So various ions, for instance, can diffuse from the cytosol into the nucleus and back. But for larger molecules, these are going to be selectively transported through these pores. So some things like mRNAs will be transported out of the nucleus via active transport. Some proteins will be transported by active transport, either into or out of the nucleus. And so that's what we call a gated transport here. Next up here is transmembrane transport. And again, if we look at the key here, transmembrane transport occurs between the cytosol of the cell and a number of different organelles, including the mitochondria and the endoplasmic reticulum. And what's happening here with transmembrane transport, this is when a protein transporter, so there's a protein transporter in the membrane of these organelles. And what it does is it transports an unfolded protein from the cytosol into these organelles. We'll look at an example in just a minute of transmembrane transport. Sure. For transmembrane transport, there's a transporter that is found in the membrane of these organelles. And this transporter will transport an unfolded protein across the membrane of these organelles and into them. And then finally here, there is vesicular transport. And again, you can see using this key here that vesicular transport occurs between various organelles in a cell. So for instance, between the endoplasmic reticulum and the Golgi, and then say from the Golgi to the cell membrane. There's some that go from the cell membrane into the cell as well. And for vesicular transport, we see it on this slide here. It's when a transport vesicle that is made up from the donor organelle's membrane. So we have this vesicle budding off from the donor organelle. We then form a vesicle, which then can be transported in the cytosol to another organelle or to the cell membrane, where it then fuses, and now the contents of that vesicle have been transported from this donor organelle, this donor compartment, to the target compartment here. Okay, so again, we have our three major means of protein transport in the cell. We have gated transport, we have transmembrane transport, and then thirdly here we have vesicular transport. One question that we can ask here is, how does each protein get to its correct location in the cell? Right? So how does each protein get to its correct location in the cell? And this is an important question, right? Because most proteins will function in a certain location in the cell. So for instance, with transcription, right? So RNA polymerase 2 in eukaryotic cells for mRNA. Does it make sense for that to end up, say, in the Golgi apparatus? No, right? So where's the RNA polymerase 2 going to be targeted to? Probably the nucleus, and maybe a bit to the nucleus for RNA polymerase 2. So getting to the right location here is important for these proteins. So again, the question is, how does a protein get to its correct location? A major part of the answer to that question is that for proteins that will be targeted to a specific location, these proteins have what we call a signal sequence that is found within them. And this signal sequence is what will be responsible for targeting the protein to a specific location. So if there's no signal sequence, then the protein will remain typically in the cytosol of the cell. And then if there is a signal sequence, well, this signal sequence is going to act like a zip code, essentially. So it's a certain code that's attached to, or that's found within the protein that then targets it to a certain location in the cell. These signal sequences are usually between 9 and 12 amino acids in length. They often contain hydrophobic amino acids, but occasionally there are some others, some positive ones as well that can be found. These signal sequences themselves can be found on, say, the end of a protein, such as what is shown here. So here they're found on the amino end of this protein. But with that being said, they may also be found on the carboxyl end, and they can also be found, in various locations within the protein as well. Sometimes, after a signal sequence has been used, if it's found on either end of the protein, sometimes it can get cut off as well, because once it's in its the protein is in its location, the signal sequence is may no longer be needed. So, what happens here with the signal sequences is that these are a specific sequence of amino acids. And there's going to be a specific sequence of amino acids that target a protein to, say, the mitochondria. There'll be a different signal sequence that targets the proteins to the endoplasmic reticulum. There'll be a different signal sequence that targets the protein, say, to the mitochondria.
So
Yes.
for this signal sequence, they work for the vesicle, the gate transport, and the transmembrane transport?
So that's an excellent question, and the answer to that is no. So right now, with the signal sequence, this will primarily target this will definitely be used in transmembrane transport. This will, in terms of vesicular transport, there's two things that take place here. For vesicular transport, sometimes these signal sequences can be used to determine which proteins are found within a vesicle that then get transported. However, in the transport of that vesicle itself from one location to another location, signal sequences are not used. And we'll talk about what is used in a little bit here. Okay? Other questions? Yes?
So these codes or whatever, they're like part of the protein, like they're already integrated in the protein.
Yes.
So like if there was a mutation, they could be like transported to the wrong part of the cell or something?
Yeah, this is an excellent point to make here. So the signal sequences are indeed just part of the protein itself. It's found within the amino acid sequence of the protein itself. So let's take an extreme example here where, let's just say experimentally, we have a protein that normally has a signal sequence, say, to the nucleus. If we were to take that signal sequence, remove it, and replace it with a signal sequence for the endoplasmic reticulum, that protein would end up at the endoplasmic reticulum. Okay, so now, back to your point here, when we have a protein with a signal sequence and that signal sequence gets mutated, and if it gets mutated enough, so that way it's not, it can't be used, then most likely that protein would remain in the cytosol of the cell, and it wouldn't be targeted to a specific location. Okay, so that's an excellent point to make in the sense that there can be mutations that do occur in signal sequences that can affect where they end up. So again, the exact signal sequence here, like a zip code, right? A zip code you put on a letter, that determines where either the letter or the protein would end up. In terms of what's going on here, you can almost think of the signal sequence providing, you know how there's like a ligand receptor interactions that can exist? Well, it's going to be a similar idea here, where the signal sequence, you can almost consider it as a ligand, and there'll be receptors for that specific signal sequence in various parts of the cell. I'll give you an example here in the next section. And for this example here, we'll see what happens with protein transport into the endoplasmic reticulum. Before we get to that, I'll just provide you a few things relating to the bigger picture here with the endoplasmic reticulum. And what we're going to be focusing on in just a second is going from the cytosol to the endoplasmic reticulum. And this is going to involve, if we went to the earlier slide here, this will involve what we call transmembrane transport. And then from the endoplasmic reticulum, the proteins will be transported to the Golgi, and then they can be transported to the cell surface, where they may be exported from the cell, or they may remain in the cell surface, the cell membrane itself. So that's the big picture for what's happening here with protein transport in relationship to the endoplasmic reticulum. The endoplasmic reticulum itself is an organelle with an extensive membrane system, and it plays a major role in protein transport, so what we're going to be talking about here, but it also plays an important role in lipid synthesis as well, among some other things. But for today, we're going to focus on the protein transport. The endoplasmic reticulum itself is surrounded by a phospholipid bilayer, and the inside, very simply, is called the ER, or the endoplasmic reticulum lumen. And then on the ER itself, on the membrane itself, there are two versions of this, two types of it. And we all know there's smooth and rough endoplasmic reticulum. The smooth endoplasmic reticulum is where the lipid synthesis takes place primarily. And the rough endoplasmic reticulum, what makes it rough? What's attached to it? Ribosomes. Ribosomes, very good. Yeah, so we have the ribosomes that can be attached here. And so since we're going to be discussing protein transport today, we'll be focusing really on the rough endoplasmic reticulum. So, yes.
Smooth as lipid synthesis.
Yes, smooth is the smooth ER is especially involved in lipid synthesis. So let's see here how a protein gets into the endoplasmic reticulum. So to begin with a protein being synthesized, right? So we just talked about protein synthesis initiation. So a couple of classes ago, we talked about this. And remember, during protein synthesis initiation, we have some initiation factors that are involved. But eventually here, the large and the small ribosomal subunits attach to the mRNA. And protein synthesis begins. Protein synthesis, or the initiation of protein synthesis, is going to occur in the cytosol, no matter where that protein will end up. But here, since we're talking about one that will end up in the endoplasmic reticulum, let's see what takes place. So we have protein synthesis initiation beginning. We then have the first steps of elongation of the new protein occurring. And you can see that with this little squiggle here. That's blue and pink. The pink, by the way, is the signal sequence in this figure. And so what happens here is the signal sequence gets produced on the 5' end of the new protein that is being produced. And once it is found outside of the ribosome, so the signal sequence, in this slide here, they call it a signal peptide, the same thing. Once this ER signal sequence has been produced, it gets bound by this particle here called the SRP, which stands for a signal recognition particle. This signal recognition particle now causes protein synthesis to temporarily stop. So there's a pause in protein synthesis. And now, this now provides enough time for the SRP, so the signal recognition particle, to bind to its receptor in the endoplasmic reticulum membrane. Okay, so now the SRP will essentially, again, pause protein synthesis, and it now leads to the binding of the SRP to the endoplasmic reticulum. And now, once bound here, the newly synthesized protein is transported to a nearby translocon, this pore in the membrane of the ER. And now once this takes place, the SRP diffuses away, protein synthesis is resumed, and now rather than this protein being translated into the cytosol of the cell, it is now being translated into the endoplasmic reticulum. And now in this case here, there's an enzyme, a peptidase, that removes the signal sequence. It's no longer needed. That protein finishes being produced, it folds, and now this protein is found in the endoplasmic reticulum. So this is what happens with transmembrane transport. So, question?
Is this happening in the rough ER?
Yes, this is happening.
Let's move on.
Right, exactly. So this is what's happening in the rough endoplasmic reticulum. So let's do a question here, and we'll do this with poll everywhere. And the question is, what do you think, true or false, the initiation of protein synthesis will proceed in an identical manner between those proteins that will remain in the cytosol and those proteins that will enter the endoplasmic reticulum? So go ahead and enter in your response. So let's see the results here. So true and false. So we have about a third with true, about 2/3 with false. Let's see, the correct answer is true. So exactly. So the key to this here is initiation. So if we go back to this slide here, these first steps here with initiation and the ribosome coming together, the small and the large subunits coming together with the mRNA, that is occurring the exact same way whether the protein is going to end up in the cytosol or the endoplasmic reticulum or, say, the nucleus. Where things start to diverge then is once this signal sequence has been produced. And once the signal sequence has been produced, this is occurring during elongation of this new protein. Once the signal sequence has been produced, that's when things can start to go different directions. So here we saw what happens with the endoplasmic reticulum. You know, a similar thing could happen, say, with the mitochondria, with a protein being produced and ending up in the mitochondria as well. But again, the main point here is initiation takes place in a similar manner, no matter where the protein is going to end up. Questions on this section? All right, well, let's talk about vesicular transport now. And earlier, I mentioned that proteins can be transported between various parts of the cell by vesicles, and there's actually a lot of this that is occurring. And notice here that we have several different pathways. So for instance, we have a secretory pathway where these vesicles can be derived from the endoplasmic reticulum, perhaps carrying the proteins that we were just talking about. Those vesicles can be transported to the Golgi apparatus. The Golgi apparatus itself has various compartments that we call cisternae. And so we have vesicles going from these various cisternae to one another. And then we have transport going, say, from the Golgi apparatus to the cell membrane. We also have some endocytic pathways, for instance, where we have vesicles forming at the cell membrane and entering into the cell. And then we also have some retrieval pathways where some of these vesicles essentially return to a location where they originated from. Okay, so we have a number of vesicular pathways, vesicular transport pathways here. Question? Yes, so now with vesicular transport, this is after the proteins have been formed. So from our discussion so far about vesicular transport, you might just assume that the vesicles themselves involve only lipids. And while lipids are important here for the formation of vesicles, there are also some proteins that are involved. And the vesicles that we're going to be talking about today are actually what we would call coated vesicles. And in other words, there's a network of proteins that surrounds the vesicles and is involved in their formation. And the three major types of coated vesicles are clathrin-coated vesicles, COP1 coated vesicles, and then COP2 coated vesicles. And overall here, these vesicles are going to do a couple of things. Overall these coats, these protein coats, are going to do a couple of things for the vesicles. First of all, they're going to play a role in helping to localize proteins to be transported. So they'll localize proteins to be transported into these vesicles. In addition, these protein coats help with the formation of the vesicles themselves. Okay, so we should know these three types of coated vesicles. So clathrin-coated vesicles, COP1 coated vesicles, and COP2 coated vesicles. Each of these types of vesicles is going to be used in certain locations in the cell. And so let me orientate you to some of the key points to this figure here. So first of all, remember on an earlier slide, we saw the different pathways. So if I go back to this slide here for a second, we see there's a secretory pathway in red, an endocytic pathway in green, and a retrieval pathway in blue. Well, those arrows are still similar here. Okay, so sometimes that can get confusing with how this figure has been produced. So, but now, for the moment here, let's focus on the coated vesicles. So, clathrin-coated vesicles form at the cell membrane and are used in the endocytic vesicular transport. You can see that they are also found in some other locations in the cell as well, but the one that is most important and the one I want you to know is that the clathrin-coated vesicles form at the cell membrane. For the COP1 coated vesicles, these ones here will form at the Golgi. And so, for instance, they'll go from cisternae to cisternae of the Golgi. They'll also go from the Golgi, they'll form at the Golgi, and then they'll go to the cell membrane. And then there's also a few that are involved in the retrieval pathway in the Golgi as well. And then lastly here, there is the COP2 coated vesicles, and these form at the endoplasmic reticulum and are then transported to the Golgi apparatus. Once the vesicle has formed, the coats will actually dissociate. Okay, so once the vesicle is formed, the coats essentially come apart. But again, for this slide here, you should know these three types of coated vesicles and the locations where they are formed and also where they end up. If I did not mention something here, you don't need to know it for, say, a quiz or an exam. So for instance, from an early endosome to a late endosome, I'm not going to ask you that a COP1 coated vesicle is involved. So know this figure at the level that I just discussed it. Okay? Pardon?
You could now.
You could now.
Mention it.
Sure. Oh, well. Now you'll have to figure out whether that will be on the quiz or not. No. Question. Yes.
Sorry, for the moving from the Golgi apparatus outside of the cellular membrane, it's coded for top 1, but it's got the mechanism arrows for top 2. What does that mean?
So, right. So let me clarify this. And this is what I was trying to clarify early on. So these arrows here, it's unfortunate that they're the same colors that they used over here, because the arrows here simply refer to the different pathways. So this is the exporting pathway from the cell.
Oh, all right.
Okay. Whereas the green is the endocytic pathway. So yes. So thank you for clarifying here, because again, these arrows here, the color of them do not correspond with the key over here. It took me actually a long time to figure that out with this figure. So yes.
All right.
Other questions? That was the same question. Okay. Yes.
The plasma and vesicles.
Pardon?
The plasma and coated vesicles that
Yep.
form the cell membrane.
Yes.
So does that stay on the cell membrane or does it go?
So good question. So clathrin-coated vesicles, these are formed at the cell membrane. And what happens here is these vesicles that are formed at the cell membrane will enter into the cytosol of the cell, where they typically will fuse with endosomes. And so this is a way, for instance, these clathrin-coated vesicles, there's a number of things that can occur with them. So for instance, they can take in some, typically if there's a ligand receptor interaction that has occurred at the cell membrane, these vesicles will maybe take that in, and that's a way to, first of all, maybe turn off this response at the cell surface. It's also a way to get some components from the outside of the cell to the inside of the cell, where they may be degraded and then used by the cell itself. So there's quite a bit that can occur here with the clathrin-coated vesicles and endocytosis.
Just as a
Yep.
coated vesicles basically direct the proteins. It tells it which way to go.
So yeah, let's clarify that. So the coats of the vesicles have 2 roles, right? So they help to determine what proteins end up within the vesicle itself for transport. And then they help with the vesicle formation itself. So your question is getting at sort of the next part that I want to talk about here, which is how do the vesicles go to where they're supposed to go? And that question there, right? So the question is, how do vesicles go where they're supposed to go? And that question is at least partly answered by these proteins called snares. And what happens here is there are 2 types of snares. There are V snares. What do you think the V stands for? Vesicle. Yeah, so we have V snares and we have T snares. The T stands for target. So these are going to be found on the target membrane. So on the vesicles, we have V snares, and on the targets, we have the T snares. And what happens now is in the cell, there are a number of different V snares and a number of different T snares. And for each of these, for each T snare, there is a particular V snare that will bind with it. So let's see how this works on the next slide. So this slide here is showing that we see 2 vesicles being formed here. Notice that each of these vesicles has a different V snare that is found on it. And what happens now is these vesicles can be transported through the cell And now, if the appropriate V snare can interact with its appropriate T snare, then that can lead to the fusion of the vesicle to the target membrane. And you can see that up on top here, and then you see an example of a different one on the bottom here. However, if this vesicle here with this pink V snare came down here and was transported, say, to this other target, this one here, this V snare, for the sake of our discussion, would not bind to this T snare. We would not get fusion, and this vesicle would carry on until it was at its appropriate location. Yes?
So the vesicles fusing to the membrane, the plasma.
Yeah, exactly. Good. Yeah, so the vesicles here fuse to the membrane of the target organelle, and in doing so, when they fuse, they're then able to release their contents into that target organelle, or that, you know, or if it's a cell membrane, it releases it out of the cell.
Are these snares only on coated vesicles, or are they on secretory vesicles?
So, yeah, so the question here is, are the snares on the secretory vesicles or just the coated vesicles? So to clarify, the coated vesicles are involved in those different pathways that we talked about to begin with. So they're involved in the secretory pathways, and they're involved in the endocytic pathways. And so these T and V snares are indeed involved in sort of determining where those vesicles go in all of those different vesicular pathways that we talked about. Okay. Other questions? All right, so now we have our vesicles to their appropriate site, and again, they were able to release their contents into their target organelle, or release them from the cell. So that takes us to the end of this section here. What I'd like to do is to now move on to this next section in the time that we have remaining, and at least provide an introduction for where we're heading. So we've now seen how proteins are produced in a cell. We see how they can be transported, and now it makes sense for a cell to maybe be able to respond to its environment. You know, maybe there's changes in the internal environment, maybe there's some changes to the external environment. This is a complex topic, I won't lie. And in graduate programs, there are entire courses dedicated to this topic. You could probably study it for a lifetime and still not understand what goes on. So we have 4 minutes and a couple of lectures. So this, yes, this is just an example of the complexity that we're dealing with. Don't shoot the messenger, okay? So this, I'm not going to ask you to memorize this, okay? I'm putting this up, you can if you want, but I'm putting this up here as an example to you of the complexity of the situation that we're dealing with. So for instance, on the cell surface, there are multiple receptors, multiple different receptors. Each of those might trigger some different signaling pathway in the cell. For many of these pathways, there are enzymes involved, and there's different forms of the enzymes that are involved. There's some cross interaction between pathways. And so it gets awfully complex awfully quickly, and there's a lot that we have learned, but there's a lot that we have to learn as well. as, say, as scientists and other people involved in healthcare. So as we go forward, I'm going to break this down to you and provide you just with a couple of windows into what's occurring here. Okay, so don't just blank out when you see this slide. Again, I'm not going to ask you to memorize this slide, but nonetheless, we'll have a couple of windows that we'll look into the cell, and we'll see what is happening there to provide us with an example and an understanding of what does happen within cells. So we'll get to that in more detail next class. What I would like to do in the remaining 2 minutes here is I want you to take a card here, and there'll be, we have some in-class activity points for this class, so you'll get some points for this. This is more looking for your participation and providing a reasonable answer. What I want you to do very simply on a note card here is to write your name, of course, and then write down what you would say might be 2 key points from today's lecture and 1 question that you have about today's lecture. Okay, so 2 key points and one question. So take some of these cards and just pass it around, please. That's probably the quickest way to get them to everyone.
No, but this whole lecture will be covered.
Yeah, pretty much, yes.
I got like that.
Yeah.
I'd probably not.
There you go. Just one. Whoa. So while you're doing that, there was a question about the quiz on Wednesday. So obviously, since I'm not quizzing you, I'm not expecting you to know this slide. This particular lecture will not be on the quiz on, did I say Friday? On Wednesday. On Wednesday. So just 20 and 21 for the quiz. Yeah, just 20 and 21 for the quiz.